Body fluid and circulation. You have learned that all living cells have to be provided with nutrient O2 and other essential substances. Also the waste or harmful substances produced have to be removed continuously for healthy functioning of tissue. It is therefore essential to have efficient mechanism for the movement of these substances to the cells and from the cells. Different group of animal have evolved different method for this transport. Simple organisms like sponges and coelacanths circulate water from their surrounding through their body cavity to facilitate the cells to exchange these substances. More complex organisms are use special fluid within their bodies to transport such material. Blood is the most commonly used body fluid by the most of higher organisms including human for this purpose. Another body fluid, lymph, also help in transport of certain substances. In this chapter you will learn about the composition and properties of blood and lymph tissue fluid and the mechanism of circulation of blood. It also explained here. Blood is a special connective tissue consisting of a fluid matrix, plasma, and foam element. Plasma is a straw-colored viscous fluid consisting nearly 55% of the blood. 90 to 92% of the plasma is water, and protein contributes 6 to 8% of it. Fibrinogen, globulin, and albumin are the major proteins. Fibrinogen are needed for clotting or coagulation of blood. Globulin primarily are in involved in defense mechanism of the body, and albumin help in osmotic balance. Plasma also contains small amount of minerals like glucose, amino lipids, etc. also present in the plasma as they are always in transit in the body. Factors for coagulation or clotting of blood are also present in the plasma in an inactive form. Plasma without clotting factor is called serum. Form element कौन सा होता है? Erythrocyte, leukocyte, platelet or collectively called formed element. Figure 15.1 and they constitute nearly 55% of the blood. Erythrocyte are red blood cell. RBC are the most abundant of all the cell in the blood. A healthy adult man has on an average 5 million to 5.5 million of RBC. MM3 of blood. RBC are formed in red bone marrow. In the adult, RBC are devoid of nucleus. The most of the mammals are biconcave in shape. They have a red color, iron containing complex protein called hemoglobin. Hence, the color and the name of these cells. A healthy individual has 12 to 16 grams of hemoglobin in every 100 ml of blood. These molecules play a significant role in transport of respiratory gases. RBC have an average of span of 120 days, after which they are destroyed in the spleen, graveyard of RBC. Leukocytes are also known as white blood cell as they are colorless due to the lack of hemoglobin. They are nucleated and are relatively lesser in number, which averages 6,800 mm3 of blood. Leukocytes are generally short-lived. We have two main categories of WBC: granulocyte and granulocyte. Neutrophil, eosinophil, basophil are different types of granulocyte, while lymphocyte and monocytes are the agranulocytes. Neutrophil are the most abundant, 60 to 65% of the total WBC are. Basophil are at least 0.5 to 1% among them. Neutrophil monocytes 6 to 8% are phagocyte cells which destroy foreign organisms entering the body. Basophil secrete histamine, serotonin, heparin are involved in inflammatory reaction. eosinophil 2 to 3% resist infection and are also associated with allergic reaction. Lymphocyte 20 to 25% are of two major types B and T form. Both B and T lymphocytes are responsible for immune response of the body. Platelets are also called thrombocytes, are cell fragments produced from megakaryocytes, special cell in the bone marrow. Blood normally contains platelet mm. The platelets can release a variety of substances, most of which are involved in a coagulation or clotting of blood. A reduction in their number can lead to clotting disorder, which will lead to excessive loss of blood from the body. Blood group. As you know, blood of human being differ in certain aspects to it appears to be similar. Various type of grouping of blood has been done. Two such grouping, the ABO and RH, are widely used all over the world. ABO grouping is based on the presence or absence of the two surface antigen chemical that can induce immune response or the RBC, namely A and B. Similarly, the plasma of the different individual contain two natural antibodies. Protein produced in response to antigen. The distribution of antigen and antibody in the four group of blood AB, and O are given in table 15.1. You probably know that during blood transfusion, any blood cannot be used. The blood of a donor has to be carefully matched with the blood of a recipient before any blood transfusion to avoid severe problem to clumping destruction of RBC. The donor's compatibility is also shown in the table 15.1. From the above mentioned table is evident that group O blood can be donated to person with any blood group, and hence O group individuals are called universal donor. Person with AB group can accept blood from person with AB as well as the other group of blood. Therefore, such person are called universal recipient. RH grouping. Another antigen, the RH antigen similar to one present in rhesus monkeys. Hence, RH is also observed on the surface of RBC of majority. Nearly 80% of humans, such individuals are called RH positive. RH positive, and those whom in this antigen absent are called RH negative. An RH negative person, if exposed to RH positive blood, will form specific antibodies and against the RH antigen. Therefore, RH group should also be matched before transfusion. A special case of RH incompatibility mismatching has been observed between the RH negative blood of a pregnant mother with the RH positive blood of fetus. RH antigen of the fetus do not get exposed to the RH negative blood of the mother in the first pregnancy, as the two bloods are well separated by placenta. However, during the delivery of the first child, there is a possibility of exposure of the maternal blood to small amount of RH positive blood from the fetus. In such cases, the mother starts preparing antibodies against RH antigen in her blood. In case of her subsequent pregnancies, the RH antigen from mother, RH negative, can leak into blood of the fetus, RH positive, and destroy the fetal RBC. This could be fatal to the fetus or could cause severe anemia and jaundice to the baby. This condition is called erythroblastosis fetalis. This can be avoided by administering anti-RH antibody to the mother immediately after the delivery for the first child.